Good morning, everyone. Welcome to Monday and to the fourth talk in our Breakfast Club series, where no matter where you are on your self-isolation roller coaster, whether you're feeling super productive and engaged or just doing really well to be sitting upright in a chair, we hope we have something for you. Our guest today is Dr. Luiz Rocha, Ichthyology Curator at the California Academy of Sciences and core member of our Hope for Reefs team. Hi, Luiz.
Hello, everybody.
His work's fascinating, his dives require actual bravery, and his photography is stunning, so I think you're really going to like this one. Before I turn it over, just a reminder that if you're watching... Oh, hello, dog.
Where is he? Right there.
Oh, all right. Well, if you have questions for the dog and you're watching live, whether on Facebook or YouTube, you can just leave them in the comments anytime at all. And at the end of Luiz's talk, we'll circle back and ask as many of them as we can. With that, Dr. Luiz Rocha, Understanding the Coral Reef Crisis. Thanks, Luiz.
Thank you. I thought I knew the dog was going to do that. I just didn't know it was going to do it before I even started, but oh well. Start things over. I hope everybody's doing okay. Crazy times, huh? I hope the talk is not very depressing. I kept changing the slides and changing because it's really hard these days to talk about coral reefs without making it depressing, but I think there's a silver lining in the end, so hang in there and it's gonna get better. So the topic today is coral reefs and the coral reef crisis and how they interact and why it matters and what we're doing about it. So coral reefs are the most diverse marine ecosystem, one of the most diverse ecosystems on Earth. One of the places where there's a really high number of species packed in a very small area. To give you an idea, coral reefs, they cover less than 1/4% of the surface of the ocean, but about 30% of all species in the oceans live on coral reefs. So they're super, super diverse, lots and lots of species, big, small, pretty, ugly. If you follow me on social media, you know I like the ugly ones. And coral reefs are in trouble for a number of different reasons. If I talked to you about this 10 or 20 years ago, I would be talking about overfishing, about pollution, something like nutrients, plastics, about physical damage to the reefs, like people anchoring to reefs. But more and more, these threats that we used to call local threats, they're giving way to a much bigger global threat. So this is a picture I took in Tahiti last year in April. We did an expedition there to explore both the shallow and the deep reefs. And back in April when we went, it looked relatively healthy. Everything was nice, super high diversity, what you'd expect from a reef, from a healthy coral reef. And we came back just a month later, a month and a week later, about 5-6 weeks later, and that's what we found. A very extensive coral bleaching, and that's the biggest threat to reefs today, is that climate change, warming temperatures are warming the waters and are causing reefs to bleach. Corals, a very high proportion of corals on the reef, which are the building blocks of the coral reef ecosystem, they're bleaching. And that happens because corals, they live in this very tight symbiotic relationship with an algae. It's an obligate relationship, so they can only live if they have this alga inside their tissue. And the alga is what gives them the color. So the photo in the left is like nice healthy corals. The photo in the right, they're all bleached because they got so stressed because of the high temperature that they were exposed to, that they expelled all of the symbiotic algae that was inside their tissue. So by doing that, not only they turned white, but they also lost nutrients. They lost their food. So a symbiotic relationship in biology is a relationship where both organisms benefit from the relationship. And many times they can't survive without one another. So in this case, the algae, it gets protection from predators. So it lives inside the coral, and it's very hard for anything else that wants to eat it because it's inside the coral. In exchange, it gives the coral its waste. So when the algae produces waste, corals use it as food. And when the coral produces waste, the algae uses as food. So the algae gets food in a home, and the coral gets food. Now, when the coral, the relationship is so intimate that when the coral gets stressed, it expels the algae. Just like when we get stressed, our immune system is compromised. When the corals get stressed, they expel the algae. And in this case, what's making them expel the algae is high temperature. So when the water gets hot, they expel the algae, they turn white because they have no algae in their tissue. The algae is what gives them color. And if the water stays warm for long enough, it causes them to bleach. They turn white. And they starve to death. So if the water comes back to normal, they can reacquire. If the water comes back to normal temperature, they can reacquire the algae and then continue on living. But if the water stays warm for long enough, they'll die. And this is what happened in the Great Barrier Reef, which is what we're hearing more and more every day, as massive bleaching events in places like the Great Barrier Reef. And we're hearing it about the Great Barrier Reef more than any other place, mostly because there's several universities off of Australia that are constantly monitoring the Great Barrier Reef. So the Great Barrier Reef is the largest marine protected area of coral reefs in the world, one of the best protected areas, and it's suffering with this time and time again. Every year there's a massive coral bleaching event. They used to be one every 10 years, then it became one every 5 years, then it became one every other year, and now there's almost one every summer. And every time it happens, it seems to be getting bigger and bigger. So this is the bleaching. The red in that picture there shows you what bleached in 2016 and then after in 2017. And we're just getting reports. This is fresh off the press. This article came out in the Washington Post today, show that our scientists that did all of those surveys back in 2016 and 17, they're showing now this year that there's even more mortality this year than there was in the previous years. This is from today. Oh, so depressing stuff. All right, so let's change the gears a little bit and go a little bit deeper on the reef. So this is what I've been doing on my lab lately. So almost everything we know about the reefs, including all of the bleaching, is studied on the top 200 feet, the top 60 meters of the reef. That's the shallow coral reefs. That's all of the pictures I showed you for until now. It's all shallow reefs, between 20 and 30 feet or so. And this is the area that it's being affected the most by bleaching and other local and global threats. But coral reefs, they extend deeper than that. They go as deep as about 500 feet or so. The species composition changes a lot, but there's still a lot of houses. There's still a lot of diversity structure for fish and other organisms to live. So it's still a very diverse ecosystem. And it remains unstudied for a long, long time, mostly because when you're studying the shallow reefs, you can use regular scuba diving, which is really accessible. Anybody can do it. And if you have the means, the financial means, the funding to use a submarine, you just don't stay in that shallow 60 to 150 meters or 200 to 500 feet depth. You just go as deep as the submarine can go. And a submarine is really not the right tool to study the taxa, the species that I'm interested in. So I'm interested in fish. I'm an ichthyologist. I'm a fish biologist. I want to study the fish. If I try to study the fish on a coral reef with a submarine, that would be equivalent to try to study the birds in a rainforest with a helicopter. It scares everything away, everything hides, and you can't see anything. So you can still study the corals, you can study the sponges, you can study the things that don't move, but the fish, they run away for the first sight of a submarine. So it's really not the tool to study fishes at those depths. So what people did in the past was this. To go to those depths, it's so complicated that you can't breathe air. The physiology interactions between the body and pressure don't allow you to breathe air at those depths. Both nitrogen and oxygen become toxic when you go past 200 feet or so. So you have to replace those gases with helium. And helium is a very, very expensive gas. So if you see on that photo on the right there, the scuba diver is using a regular scuba gear equipment, a regulator, and is breathing in from the tank and then expelling the gas. That's all helium coming out because they're very deep. So that would be a super, super expensive dive, because every time you breathe like that, it would be a $5 exhale because helium is so expensive. So the solution for that is using rebreathers. Rebreathers are a type of equipment that allows us to recirculate the same gas over and over and just add the oxygen that we need. So as we exhale, the gas that comes out of our lungs goes back into the back of the system. There's a filter that removes the CO2. There's oxygen sensors that constantly monitor the presence of oxygen and then add only the oxygen that we need. And then the same mixture comes back again. So we're breathing basically the same helium over and over again, removing the CO2 and adding oxygen. That makes this system very, very efficient and allows us to explore those depths. So one of the first things we wanted to do was to test this very well-known and accepted idea that those deeper reefs, because they're so hard to reach, they are somewhat of a refuge for the shallow species that are suffering so much. And that accepted hypothesis comes from those two premises there. One, that everybody thought that there was very high species composition overlap between shallow and deep reefs. In other words, everybody thought that the species that were in shallow reefs were the same as the ones that were on deep reefs. And that because they're out of diving reach, they would be also out of a lot of human and natural impacts. So we set out to do that. And one of the first things we did was try to look at how the overlap in species between shallow and deep was established. And it was established using depth ranges. And that's very, very interesting, because that's really not a very precise way to determine species overlap between two habitats in the ocean. Mostly because depth ranges, they include all of the historical records to make a composite of a species depth range. So here is one of the most popular websites that you can go and get information about fish. All of the information is very scientifically accurate and includes the depth range, and that's where most scientists would get the depth range if they wanted to study that fish, and if they wanted to check if that fish is a shallow fish that uses the deep reef as a refuge. Now, if you look at the last line on the bottom there, it's probably small letters. I mean, it's small on my screen. It's probably bigger in yours. But it says there, right there, marine reef associated on the bottom, depth range 18 to 63 meters. So that cutoff between shallow and deep, more or less, depending on what publication you look at, is at 30 meters. So if that species goes from 18 to 63 meters, it goes into that database that says that the species is a shallow water species that occurs on deep reefs. Therefore, it's a species that can use the deep reefs as a refuge. But when we actually go to the reef and we dive using all of that complicated system, the Rebreather, we see that 99.8% of those fish are found between 50 and 60 meters. So even though once, I don't know, in 1925 or something, someone collected this species at 18 meters depth, most of its population is between 50 and 60 meters. 99.8% of its population is between 50 and 60 meters. So it's exclusively deep, and it's not a shallow species that uses the deep reef as a refuge. And it's the same thing for other, not just fish, for other taxa too. So this is, these are two of the most threatened species of coral in the Caribbean, the elkhorn and the staghorn corals. Their official depth range is 1 to 60 meters. So again, if you're looking at corals and you look at that database with all of the official depth ranges, you think that this species is a shallow species that uses the deep reef as a refuge. But when we actually go to the field, we see that 99.9% of the individuals of those species are between 0 and 10 meters. So what that tells us is that most of those individuals are actually in shallow water, and there's very few odd ones that are in deep water. They're probably not going to be able to help the shallow population in any way. So we did a lot of that over and over again, several transects, to try to understand how many species actually can use the deep reefs as a refuge. Now, the other assumption there is that those deeper reefs are out of reach. They're out of reach from humans, so they're not impacted, so they're this pristine area. And that comes from the fact, basically, that we don't see them. We don't explore them. We don't know what's going on with them, so we just assume that because we don't dive there, people can't reach it, so there's no impact. So we tested those two things: the impact, and two, the overlap of species. To test the overlap of species, we did what Hudson is doing in that picture there that I took in Ponape. That was at about 460 feet depth, so that's 130 meters or so depth. So we do transects. We lay down those transects that are 20 meters long, and we count every species of fish along those transects. We counted more than 50,000 species of fish. We took a whole bunch of photographs of corals and the bottom throughout the world, and we analyzed all of that data. And during that process, we started finding something very interesting. We started finding new species all the time. So these are some of the species that our group and our colleagues from Hawaii, from the University of Hawaii, Rich Pyle and his group from the Bishop Museum, discovered over the past few years or so. And there's some spectacular looking species that nobody has ever seen before. So there's a few different ways where people find new species these days. One of the ways is cryptic species. So it's one species. One thing that was described as one species sometimes gets split into several different species, because now we have new tools and new technology to understand, to see if they're different or not. For example, giraffes were recently split in different subspecies because their DNA looks different. But if you look at them for an untrained eye, they all look the same. Now, these species that we're finding, for the most part, these species that we're finding on those deep reefs, they are really different from everything else we've seen before. They're very stunning. They have those bright colors that we're still trying to understand why. And everywhere we go, every time we go past that 60-meter, 70-meter threshold, which is where scientists more commonly dive, we start finding new species. So here's an interesting table that Rich Pyle put together, and I started adding stuff to it. Those are expeditions that both of our groups did over the past few years or so. That's the number of new fish species that we found on those expeditions. And Rich came up with this idea of calculating how much time it takes to find those species. So those deep dives, we're exposed to such high pressure that we can't stay a long time down there. We can't stay much further than 10 or 15 minutes. So we have only 10 or 15 minutes to catch everything we can. And because there's so much gas that gets inside our bodies, gets diffused into our tissues, we can't come up fast. We have to come up very slowly. So it's equivalent to a soda bottle. So when you shake a soda bottle and you open it quickly, all of those bubbles that are diffused into the soda, they come out of solution very quickly, and then the bottle really explodes. So it's the same thing that would happen to our bodies if we were to be exposed to that pressure and come up very quickly. So we have to come up really slowly. So for a 10 or 15-minute dive, it takes us 5 or 6 hours to come up. So the exploration time there on that table is only those 10 or 15 minutes. So it's only the time that we spend at actual depth collecting those species. And it varies depending on the expedition and how much time we spend in the water and how many divers we have. It varies between 1, 2, 3, 5, 6, 8 hours of exploration per expedition. And the really interesting number is this one. It's the new species discovered per hour of exploration, which on average is more than 5, which is just stunning. So we have, our group has been consistently describing 5 or 6 new species per year, each one of them more spectacular than the other. And it's just mind-blowing to me that there's still so many new species to be discovered and such an unknown habitat. It's not just new species, though. It's also new records. So everywhere, sometimes those species, they're caught by hook and line, and somebody described them 10, 20, 30 years ago because they found one in a fish market, and it's never been seen again. So a lot of those species are described from one place, and everybody thought they were just from that one place. Those species in that slide there are all what we call new records. So they're described from one area, and then we find them in another area. So they're unique to those deep the deep habitats, and they have their distribution much wider than we previously thought, mostly because nobody's exploring it. So every time we explore a new place, we found species that were described from somewhere else, and we find new species, new records, new species. So novelty all over the place. I literally feel like someone going to a new planet for the first time and seeing all of that novelty right there in front of them. So we summarized kind of all of that data in this graph. Are they a refuge? How much overlap there is between those depths. And the numbers that you're seeing there, they're high proportions, but that's dissimilarity. That's not similarity. So that's how different they are. So if you look at the shallow reef and compare it to the upper Mesophotic between 30 and 60 meters depth, they are between 87 to 96% different. So that's how different they are. And the deeper you go, the more different they are. So the bottom, the lower Mesophotic, when you go past 100 or 90 meters or so, it's 99.6 to 99.7% different. So almost no species overlap between deep and shallow. What about the other thing that we talked about? What about the impacts? How impacted are those deeper reefs? So this is an interesting expedition that we did with Ocean X. We went to the Bahamas with them a few years ago, and we almost had to cancel this expedition because there was a hurricane that went through the Bahamas, and it was supposed to come right when our cruise through the Bahamas was starting. So we almost had to cancel the expedition because of the hurricane. But then the hurricane got a little bit faster, and it passed through a few days before we were supposed to go out. So the captain of the ship kept telling us, well, you're not going to see good reefs. Are you sure you want to see? Because the hurricane just passed there. And I'm like, yes, we need to see this, because nobody has ever looked at those deeper reefs right after a hurricane like that. People look at shallow reefs after hurricanes all the time, but nobody is there at the right time with the right gear to look at the impacts of hurricanes on those deep reefs. And what we saw was this on that picture. This is a Mesophotic reef in the Bahamas. It's a deep reef in the Bahamas. The picture was taken at about 400 feet depth, so 120 meters depth. And the reef is covered by sand. So the hurricane, when it passed on the shallows, the waves that it generated broke a lot of the shallow water corals. But it also stirred up a lot of sand in the shallows, and that sand cascaded down on the reef and smothered it, covered all of the living organisms on the deeper reef. The sponges, the corals, the soft corals, the gorgonians, everything was covered by the sand. And because there's a lot less currents, there's a lot less energy in those deeper reefs. So most of the surface currents, the stronger surface current, the stronger currents in the ocean, they are driven by wind. So they're surface currents. The deeper Mesophotic reefs, they're not very exposed to those stronger currents. So it's harder for those particles, for the sand that gets on them, to be washed out by the currents. So they get smothered, and they are more susceptible to this kind of damage than the shallow reefs are. So there's natural impacts to those deeper reefs. We also saw a lot of bleaching, which is something we didn't expect. Bleaching and coral disease and corals dying on those deeper areas. We thought that was much more prevalent in the shallow reefs. But as it turns out, it's not just shallow species that suffer with high temperature. So yes, the deeper you go, the colder the water gets. But what matters to the corals is not the temperature itself, but it's the variation in the temperature. So even though those deeper corals, they are in colder waters, the average temperature of where they are is increasing, and it's causing them to bleach too. So it's not the same X temperature as in the shallows, but it's growing by a factor almost as much as in the shallows, and it's causing them to bleach. We also saw trash everywhere. This is a sea urchin that has an interesting behavior of attaching seaweed and shells and algae and things that it finds on the bottom, attaches to himself to camouflage itself, to look like it's a piece of the bottom. Right there, it's attached to a fishing line, and it has a piece of plastic attached to it. And this photo was taken at how deep was it? 450 feet or so. So very, very deep with lots of trash in the ocean, so much so that the urchin picked up trash to camouflage itself. And it's doing a good job because there's so much trash around it. So trash is prevalent everywhere in those deeper reefs. Fishing debris, these are anchor lines and fishing lines in Palau, and that's at about 350 feet depth. So we see these kinds of impacts that we're seeing in the shallow. We see that they extend much deeper than what we thought before. And I'm going to give you here an example of one of the most striking examples we've seen so far. So this is reefs in Guam. This is part of one of my students' master's work, Tyler Phelps, and he's studying how the Mesophotic reefs are impacted in Guam. And what we saw there was that the deeper reefs, they were almost as intact, almost as nice as deeper reefs on other areas that we visited. So they were relatively okay. They were diverse, they were colorful, there were species that we expected to see there. We saw them all. But when we got to the shallow reefs, the shallows were barren. Almost all of the coral we saw was dead. So much so that even the structure that they built was eroding away, because that moment the moment they die, they stop growing, they get eaten by something else. So there was a lot of impact in the shallows, and none of those deeper species were coming up to the shallows to reseed the shallows. Mostly because they're used to the deeper water, so they don't want to go to the shallow waters anyways. It's not their habitat, it's a different habitat. So Guam is one of the most impacted reefs in the planet as that I have been to, that anywhere has been to, like anybody has been to, I think. There's very few other reefs that are as impacted as Guam. It's a small island with a very large population. Everybody wants to go to the reef for one reason or another, either for food or for fishing or for fun or for diving. And what we see there over and over and over is those reefs that are completely barren, dead. There's a lot of overfishing, and it causes all of the shallow reefs to die, and they're not being helped by those deeper reefs. So they're really, really not a refuge. So let me tell you a story that kind of summarizes everything in one trip that we did. So this is the trip that we did to St. Paul's Rocks back in 2017. It sounds like so long ago now. St. Paul's Rocks is a tiny island. I took this picture from the tallest point in the island. If I turned around and took the same picture looking facing the other way on the island, it would be just blue water. So it's a small island with just that one building. That one building is a research station. Sometimes it's manned by Navy personnel. Sometimes it's with scientists. We again partnered with Ocean X. We took the Alucia to that expedition, mostly because we needed the support for the diving, and we couldn't get in that tiny research station. And we did our usual. So we did our transects. We counted the fish. We took pictures. We did all of our surveys. And Christina, the next slide is the video. So I'm going to show you a video here. Put your sound up, or if it's already up, but if you're annoyed with my voice and you lowered it, it's the time to put it up again. So when Christina hits play there, in a few seconds, she's going to pause it. Go ahead and hit play, Christina. Pause it right there. So that's Maradius Bell. He's our dive safety officer. He's showing his dive computer right now, and the dive computer gives us a lot of information. Those three numbers on the center are the most important numbers for us that are diving with a rebreather. That's the oxygen partial pressure. So that's 1.20, 1.23, 1.21. That's how much oxygen we're breathing, and we need to know that constantly because if anything fails, it's fatal at those depths. I'm not exaggerating here. So what the rebreather does is it automatically keeps the oxygen pressure at those values there, at that concentration. But it's a computer, and we're diving at 419 feet. That's the number on the top left corner of the computer there, 419 feet. So if the water gets into the system, everything dies, and then we have to run the whole thing manually, which we can do, but only if we know what the oxygen is. So that's one of the most important pieces of the whole equipment that we use, a dive computer. That's the depth, 419 feet. And I'm going to ask Christina to hit play again there. And that's me right there under that other light. So that's me right there. I'm catching a fish, and look at what comes up from the depth. So Meridius is saying, if you're not understanding, he's saying, look at the shark, look at the shark. Look at the shark. And I'm collecting these beautiful new species. So I'm turning to Meridius and I'm saying, I didn't understand what he said. And I looked at him and I said, I'm okay, wait, I'm catching this fish. I don't want to go up now. And then he's yelling at Hudson now. Hudson turns to him and says, I'm okay, everything's fine, I'm catching this fish. So hit pause again now, Christina. So neither Hudson nor I saw the fish, but here is how the whole thing comes up in context. So we're catching these new species that nobody has ever seen before. We're at 420 feet in this island in the middle of nowhere. The island is between Brazil and Africa. It's about 700 miles offshore. There's only one research station with 4 people in the island, and that shark, can you point to the line there, Christina? There you go. So that shark has a fishing line attached to its mouth. So that shark is a deep water species. It's a 6-gill shark. It never comes shallower than 300 feet or so. Certainly not at those latitudes with where there's warm water on the surface. And it already has signs of human impact right there, a fishing line attached to its mouth. So hit play just so that we can see the rest of the video. The helium voice there in Meridius is the funniest thing of the whole thing there. And then the shark swims away. So the moral of the story there is that neither Hudson nor I, both of us are together right there, neither Hudson nor, that's how much gear we take. Can you believe that? It's just crazy, but it's rewarding because this is why we do it. Can you go back to the presentation, Christian? There you go. This is why we do it. So that's the fish we were catching when we were ignoring that shark. Incredible looking new species. We named it Tosanoides Aphrodite over the Greek goddess Aphrodite because, I mean, we were so distracted by it that we ignored the huge shark swimming over our heads. So novelty, everything is different. It's a unique ecosystem. And these photos were all in St. Paul's Rocks, in the middle of nowhere. Again, 4 people in the island. And we saw in every dive, every 5-10 minutes that we spent on the bottom, we were either looking at these species or looking at trash. So there was trash everywhere. There's a pipe there, there's fishing line, there's some sort of t-shirt, I don't even know what that is. All kinds of trash that we saw in this one of the most remote islands in the world with only 4 people inhabiting it. So human impacts go far and wide and deep. So to conclude this portion, they are those deeper reefs, the Mesophotic ecosystem. Twilight Zone is not a general refuge. It can be a refuge for a few select species that do span the depth range, but for most species, it's not a refuge. The communities are very different. The species composition is very different. And a lot of those Mesophotic ecosystems, a lot of that Twilight Zone, a lot of those deep reefs, they're almost as impacted as their shallow counterparts. So now let's talk about something a little bit more positive. So all of this work that we have been doing in the Academy for the past 4.5 years are part of this 5-year Hope for Reefs initiative that Bart Shepard, myself, and Pim Bungertz and Rebecca Albright are kind of pushing along the way, along with other coral reef biologists at the Academy, including Terry Gosseliner and Gary Williams and Rich Moy. We've been going all over the place studying coral reef ecosystems, deep and shallow, throughout the planet. And one of our questions we're asking is this: is there hope? After all of this that we're seeing, all of these massive bleaching events, everything dying deep and shallow, is there any hope? And the answer to that is yes. This picture I took just last year. It was our second to last expedition. We did one last October to Brazil, to Fernando de Noronha. Before that was the Marshall Islands. So this was the Marshall Islands expedition in August of 2019. And we found giant, massive coral heads inside the lagoon. Outside of the lagoon, the corals were not doing that great, but inside the lagoon, they were. And the reason for that is because inside the lagoon, because the lagoons on atolls and coral reefs in general, they have less water circulation than the outside of the reef. So in the outside of the reef, the temperatures are much more stable. Inside, in the lagoon, they fluctuate a lot more. So because the corals that live inside the lagoon, they are already used to that higher fluctuation in temperature, they don't suffer as much with the global increase in temperature. So some species in some places are surviving. There is hope there. And the hope is that they can reseed the other reefs outside at some point when things start becoming to go normal. With that, I wanted to say that there's just one solution to this, which is slowing down and stopping and reverting global warming. We have to bring the temperatures back to normal. Otherwise, a lot of species are going to go extinct. And I don't know about you, but I don't want to be the species responsible for it. So in addition to those corals that live in lagoons that are more used to this, there's also corals that live on the fringes of their distributions. So in the colder areas, so corals, they like warm water, they like the tropics, but they extend sometimes to the subtropics, to some places like Japan, or in this case, in the picture there, this is Easter Island. So we did an expedition to Easter Island in 2016, and we saw practically no bleaching. Easter Island is so far south that the temperatures are more buffered against warming there, so they're not warming as much. And another thing that really gives me hope is the resilience of coral reefs. So this is the largest nuclear explosion ever created by man. It's one of the tests that was done in Bikini Atoll. It's a huge, massive nuclear blast in the wall of the coral reef in Bikini Atoll in the Marshall Islands. Don't even get me started with what this caused to the population in the Marshall Islands and why the US did that. That's just a topic for a whole nother talk. But the fact of the matter is that I came back there just 10 years ago, and what I saw were thriving, healthy, stunning looking reefs. And the reason for that was because we left it alone for 50 years. So after that was only the bigger test, but there were many other nuclear tests that were done in the island, and there is still high background radiation in the island, so much so that they don't allow anybody to live there for a long time. They don't allow any fishing there. So because there's no fishing, there's no impact, there's nobody on the reef. The reef was left alone for 50 years. It recovered. It recovered from repeated nuclear atomic explosions. So if it can recover from that, it can recover from everything. We just need to give it a chance. One of the things we try to do with Hope for Reefs is provide data to management agencies throughout the world. We help create, support the creation of marine protected areas. Hudson Pinheiro, one of our postdocs in the initiative, helped lead the way to create this protected area in Brazil. This is Martin Vaz Island, one of the most beautiful places in the world. It's uninhabited. It's very far from land, but there was fishing going on there. And now the protected area will give a stop to that. We also partner with SECOR. SECOR is a very interesting, it stands for Sexual Coral Reproduction. And it's a very interesting foundation that helps restore sexual coral restoration, helps restore corals through the Caribbean, which is one of the hardest hit areas. And I think Rebecca Albright is going to give a talk about this in the near future, so I'm not going to go into too much detail. But suffice to say that we're seeing coral restoration as a way to try to help some species survive while we get a handle on climate change. So I think even if we stopped climate change today, there would still be lingering effects for 50 to 100 years. So to bridge that gap, to help coral survive the impacts that we're causing now, we're looking at active coral restoration to try to keep some populations going so that they can recover in the future when the conditions come back to normal, or when they adapt to the new conditions, if that's the case. Something else we do is we have fun with taxonomy. So taxonomy is a very hard science to describe its importance to the public, but it's a science that attracts enormous public attention for many different reasons. One of the biggest ones, I think, is the thrill of discovery. So every time you announce something that nobody has ever seen before, it's attractive in one way or another. So we are having fun with not only how spectacular those species look, but how we catch them, and sometimes having fun naming them. So our new species, thanks to the media team in the academy, Christina and Laurel included, our new species have huge presence in the media, social, and mainstream media sometimes. This is that species that I showed from St. Paul's Rocks that we named Tosonoides Aphrodite. And it's interesting, there were two different waves of media around it. One was focusing on the species that was so pretty and it was neon colored, and we named it Aphrodite. And then a few days later, there was a whole boom of other media articles that were focused on the fact that we ignored that shark when we were catching it, and everybody was having fun of us talking with the helium voice during its discovery. Something else we do with taxonomic names is we name them after popular pop culture kind of things. Some taxonomists are very purist, and they don't really like doing that because they say that a pop culture today won't mean anything in 100 years or so. And I kind of agree with that, but I think that it means something now. And we named this species that we discovered in Zanzibar, in Tanzania, we named it Wakanda because of its color. So it's purple colored. It reminded me of Wakanda. One of our partners from Australia, from University of Sydney, helped us name it. And he gave the idea of putting the name Wakanda. And when he said it for the first time, I'm like, yes, let's do it. Mostly because I knew it was going to attract a lot of media attention. And whenever we attract media attention to those species, we make people think about them, and we make people care about them, and we make people create marine protected areas. We make people protect them somehow. So it's something that I think is really important to be done, is to publicize new species discovery as much as we can. And we've been doing that a lot. And then there's our public floor, which is closed right now, sadly, but I invite you to come back as soon as we reopen. We have a whole exhibit dedicated to those mesophotic coral ecosystems, and we have a lot of shallow coral exhibits on the public floor, and we can help you understand everything there is to know about coral bleaching and coral reef biology. Everything we know about it, we can talk about it in the public floor. And we have a lot of online resources to help you understand that too. So there's a whole planetarium show that we have, Expedition Reef, that has a lot of teaching resources on our website. And if there's a question about that later, I can link you to that too. But yeah, there's many resources we can point you to if you want to learn more about coral bleaching, the coral reef crisis, mesophotic reefs, Expedition Reef, all of our resources that we're using to help coral reefs throughout the world. And I want to finish by saying that this is not a sole effort. There's a lot of people that are with me on this initiative. For the mesophotic work in specific, Hudson, Tyler, and Bart are my partners in crime. Bart Shepard is the director of the Steinhardt Aquarium that always comes with us, and I kind of pulled him away from the public floor, and we're writing a lot of papers together. My wife, Claudia, and my kids, Sophia, was the one that showed me that Washington Post article today. I knew it was back in the media, but I didn't know it was coming out today. So she showed me that, and I added it to the presentation. So thanks everybody, and I'll be glad to answer questions if there are any.
Hi, Louise. Thank you. And yes, there's a lot of questions. That was fantastic.
All right.
Yeah. I should say too that so we'll work with Louise later to drop links to any resources that he mentioned into the comments section. And yeah, we are working with Dr. Rebecca Albright to get her on as well to talk about coral reef restoration. So this is like some nice peer pressure, Rebecca.
Yeah. I tried to stay away from it because I knew she was going to do a much better job than I did.
Yeah. And now she's like extra obligated. So first question from Sarah. Roughly what percentage of deep reefs have been explored by divers using rebreathing devices?
By divers using rebreather devices, I don't know, less than, I don't even know, it's such a big fraction that I don't even want to put a number, but less than 1% by a long shot. So there is a few teams that explore it using rebreathers to about 60 meters, 200 feet. So between 30 and 60 meters, I would say it's the bulk of the Mesophotic studies. Our team, along with the Rich Piles team in Hawaii, are the only two teams in the world that are doing this exploration much deeper than 60 meters. We go up to about 150 meters, that's 500 feet. So there's less than 10 people in the world, 10 scientists in the world doing this to the depths we are doing. So yeah, it would be much less than 0.1% or less.
Right. You used to use that great line for a while about, I think it was that more people had been to the surface of the moon than to the deep.
Right. Yeah, more or less scientists, definitely, to those deeper depths for sure. There's other people that do it for fun. I can't imagine myself doing it for fun, but there's quite a few people that do deep diving using the same gear that we do, going into caves and the ocean, go deep just to have a number in their computers, which is not my cup of tea. I chase fish, not numbers. Bart coined that phrase, and it works well for all the team.
Say it again.
I chase fish, not numbers.
Yeah, that's a good motto. We have this question from Scott that's kind of related, I think. He asks, how do you think about the risks versus the rewards when you dive? What's your mental strategy for tackling that every time?
Right. I wish Mauritius was here to answer that, but he's our dive safety officer, and it's always a tug of war between me and him because he wants to put more safety protocols, and I want to put less because I want to get in the water quickly and I want to catch my fish and I want to come back. But he's right. The more layers of safety we have, the safer we are. And I think the way we're doing it with the many safety layers of that we do in our dives. We have multiple checklists that we go through to assemble our gear before we jump in. We have multiple different scenarios. Every expedition, the first dive that we do is to 20 feet. Mm-hmm. And we share air, and we take our rebreathers out of mouth. We take the masks out. We kind of pretend that we have no oxygen. So what do we do if we have no oxygen? What do we do if we have too much oxygen? So we run through all of those scenarios to build muscle memory and stay current and have the best possible safety. And so yes, I'm not going to tell you that every single time it's fun. Sometimes it's tiring. It's a lot of work, especially to the build up to get to the first dive, because we have to work in addition to all of the safety protocol. There's also getting there and putting all of this gear in the plane and going through customs and getting all of the collecting permits and establishing local collaborations and doing all of that. It's very stressful. When we get to the point to do the first dive, we're almost like looking at each other and asking, why do we do this again?
Right.
And then at the end of that first dive, we look at each other and say, that's why we do it again.
Yeah.
Because every dive is a reward. So it's very risky, but I think we do it in a way that eliminates a lot of the risks. Not all, but I'm very confident that we can go and come back.
Right. And I'll ask, let's say, that we obviously get a lot of questions about that just because it's such an extraordinary approach, but I'll scatter them kind of throughout. One last one just before we move on to some reef stuff. Will asked what the certification requirements were for diving as deep as you do Yeah.
So the certifications themselves are not a lot of work. My certification is called Extreme Exposure. So there's 3 levels of rebreather diving. First, you learn how to use the rebreather using air, not using helium. And that takes about a week. And then you have to log hours, just like you do when you're learning how to fly a plane. You have to log a certain number of hours before you can fly it by yourself. And you have to log a certain number of hours before you can fly a bigger plane. So it's the same thing. We have to log a number of hours, not only during the course itself, but also after you finish the first level of training that allows you to dive to 100 feet. You have to dive for 50 hours to do the second level of training, which allows you to dive to 200 feet.
Mm-hmm.
And then you do the 1-week course to learn how to dive to 200 feet. And then after that, you have to dive for 50 hours to 200 feet to be able to do the third level of training that allows you to dive to 300 feet and beyond. Mm-hmm. So it's a lot. It's 3 levels of training interspaced by 50 to 100 hours of just getting used to using the rebreather. And I can tell you, even when we follow all of those hours, we did all of the protocols, we did all of the training the way you're supposed to do. And now I have almost 1,000 hours of diving on the rebreather. And I think I'm finally past the point of being too, I think the most dangerous area, not only for rebreather diving, but also for normal recreational scuba diving, is when you're between 100 and 300 or 500 hours of diving or so, which is when you have some experience, but not enough experience. So you have some experience to be dangerously risky. So you think you know more than you do. So now so many things happen to me, that including computers dying and rebreathers failing and me having to run the thing manually the whole time. But I think I'm past the time that I think I'm comfortable, and now I'm actually comfortable. And that's 1,000 hours of diving.
Right. And you're still, it's, I think, worth pointing out, like, you're still, you know, before each dive, kind of rehearsing all of the fundamentals that, like, every scuba diver will know from just the most basic recreational stuff. Like, you start there.
Absolutely.
Yeah. Okay, Jay Gomez asked a question about bleaching. He wanted to know, how long will corals survive without algae?
It depends on the species. It's about 4 to 6 weeks, so it's a short period of time. So not much more than a month, depending on the species. Yeah, we've seen recovery in some places. We've seen total catastrophe in other places. In Moorea, that picture I showed where there was a lot of bleaching, towards the end of that expedition, we started seeing algae colonizing or growing over the bleached coral, which is a sign that the coral died and that the skeleton is being colonized by other organisms.
Right.
So maybe a month, depending on the species.
Okay. And Bob had a question. I'm going to kind of try to sum up. Basically, he was curious about why you were talking about a lot of your findings in terms of ranges instead of averages, so a depth range as opposed to an average depth. And he's kind of wondering if is it advantageous? Are there different frameworks in which at one point it's useful to say it's found at this range versus it's found at an average depth of?
Mm-hmm. I think it would be more precise to put the average depth. The problem is that data is very hard to get, and it's not synthesized in any way. It's not summarized in any way. So you'd have to go back to every collection. We have a collection at the Academy with 250,000 jars of fish. I would have to build that database in a way that would allow me to calculate that average depth. I would have to know how many fish were collected in each depth. And in a lot of times, the collections are biased. So I just said that the shallow reefs we collect more in the shallow reefs than we do in the deeper reefs. So there would be, even if it's a species that is more common on the deeper reefs and less common in the shallow reefs, I would have more of them from shallow reefs because we collect so much more in the shallow reefs. So if I use that data and average it, it would look like it was shallower than deeper too. So it's a problem of the data not being there. And even if it was there, I think it would be problematic too, because we have the collections are not biased. If we collected the same amount in every habitat, that would make complete sense, but we don't. There's easier places to collect than others. So.
Right. And if you were, I wonder if you were talking about it in a conservation framework, might it also be useful to talk about a depth just in terms of like having people understand it as a chunk of space that needed protecting?
Right.
Okay. Let's see. So Amy asked, she says, I had no idea that you could hear each other underwater. How does that even work?
Yeah. After sometimes, after 100 hours of diving or so, we start understanding each other.
But you're basically just kind of screaming as loud as you can.
We do, yeah. So with the normal scuba gear, nobody even tries to do it because there's very little air inside the regulator, the normal scuba regulator. But with a rebreather, it's a much wider space of air that you have in front of your mouth. So the echo in there makes the sound propagate a lot easier. And the fact that we're breathing helium with the squeaky voice, that also travels in the water a lot easier than the deeper voice if we had just normal, if we were breathing normal air. So it's the fact that we're using the rebreather and that we're breathing helium makes it easier to understand each other underwater.
It's sometimes. It's like one of the best things about Mesophotic videos is you've got like this very serious work happening with like angry chipmunk, like explosions of dialogue. It's really good.
Yeah.
Okay, let's see. So Kent asked, what are the current limitations that you encounter when doing research driving with rebreathers? And what development in rebreather diving related tools are you hoping to see for future Mesophotic research?
Nice. That's a good question. I think the biggest limitation we see is that a lot of the rebreathers, they're very complex. There's too many moving parts. There's too many places where it can fail, and it does fail. So we know it fails. That's the trick there. In terms of technology, so there's a few things that we can't really monitor, like, for example, CO2. So if that filter that we have in the back that removes the carbon dioxide, which is the byproduct of respiration when we exhale, if that filter stops working for some reason, so if we're breathing too much and we're swimming against the current and we're producing a lot of CO2, sometimes there's what we call a breakthrough. So CO2 can pass through the filter and you can start breathing it again, like you're breathing in a bag. So that's not good at 500 feet or any depth, for that matter. But it becomes more prevalent the deeper you go because the gases get denser. We have no way of identifying that. There's no sensor for it. So there's sensor for the oxygen, but there's no sensor for the CO2. Mostly because nobody could come up with a sensor to monitor that in a wet system or relatively wet system, high humidity system. So a technological advance that would help us a lot would be a way of monitoring CO2 in your gas. Another one would be more reliable oxygen detection / oxygen addition. So the way we add oxygen to the system is by using a solenoid, and there were divers that died because the solenoid got stuck open and they couldn't close the oxygen in time. So it's a mechanical solenoid that's susceptible to failure. One of the things we do that we try to avoid that is having it. You're supposed to service your rebreather once a year. We service it after every expedition. So all of our dive officers, Mark, Marina, all of the other, Allison, all of the other dive officers that we have at the academy, they're all rebreather divers. Not only that, they're all rebreather technicians. So they can do the servicing on the rebreather. They do it every 3 months or so because we do on average 4 expeditions a year. What other technological advances? Maybe better oxygen sensors. So right now, the rebreather we have, it has 3 oxygen sensors. And the reason for there being 3 is because we can't trust one. So we basically use a voting logic. So if there's 2 of them that are telling us one thing and one is telling us another, we kind of suspect that the one that's different from the other 2 is wrong. But it happened with me that the 2 were wrong and the one was right. Wow. And there's ways we can find that out underwater, but it's not an easy thing. But that causes the computer to add the wrong amount of oxygen to the system. So very, very complicated.
Right. And every single thing that could go wrong has really, really significant consequences, obviously.
Oh, yeah.
And one other thing you referenced just now briefly, but I don't think mentioned in the presentation that's really interesting is just the fact that you not only have a very limited amount of time at those lower depths, like 10 to 15 minutes when you go really deep, but you also have to work very slowly so that you're not like
you can't overexert yourself. And we have so much gear attached to us that it's very easy to overexert myself. I mean, I've done it a few times.
Yeah.
When we get to the bottom, sometimes there's a current and we have to swim against the current to get to the place that you want to be. And when you get there, and the whole world goes, woo, and then you have to hold on to a rock and then breathe for 30 seconds, which seems like an eternity at those depths.
Yeah.
And then when your breathing goes back to normal, then you can kind of start working again.
Yeah. That dovetails nicely with Mari's question. She asked, so she was struck by just how much gear was hanging off of you in the shark video. And she asked if you could walk us through what all of that is and also how much does it weigh on dry land?
Right. So on dry land, on a wet day, I weigh 180 pounds. My gear I don't weigh that much. I weigh 170 pounds. My gear weighs 200 pounds. Well, sometimes more than 200 pounds. So the tanks that you see on my side on the video are bailout tanks. In case now that you know that the rebreather does fail. It's not a matter of if, it's a matter of when. So one of the things we can do when there's a catastrophic failure on the rebreather is breathe off of those tanks. So those are tanks that are emergency tanks that we only use if the rebreather completely fails. And it's so much gear because we have to take enough gas to take us back to the surface. And we have 5 hours ahead of us, so we have to have enough tanks to take us back to the surface. Another big piece of equipment that we take is a scooter. It's a dive propulsion vehicle, and we take that specifically because if there's a current, we don't want to swim against it. So we just point the scooter to the direction we want to go, and then we kind of push the trigger, and the scooter takes us.
Yeah. And it's interesting too that like your configured, like your bailout stuff is configured for you as a team, or at least it was last time I checked in. I don't know if your methods have changed.
Not anymore.
Oh, you're all completely self-contained. Cool. Meridius in action.
Exactly, Meridius in action. So back in the day, a few years ago, we used to have enough gas for one person between two people. So you and my buddy would have enough gas for one of us, or the two of us together would have enough gas for one of us to come back to the surface. And that would be okay, I guess, in a scenario where everything is perfect and you don't get separated from your dive. But if there is a catastrophic failure and my buddy is 10 meters from me underwater, do I swim to him or do I go to my bailout and go up? So that's the decision I would have to make if I had a team bailout scenario. Now that I have all the gas I need with me, I can just switch to bailout, go up, and then hope they see me, because I'm not waiting for anybody.
Okay, I'll ask you 3 more questions. I know we're going long. Thank you for staying with us, Luiz.
All right, I'm in quarantine. Look at this.
Yeah, you're busy. Brian asked, and so I think he was responding to one of your photos of just lots of trash swept up on one of those deep reefs, how it's getting there. And he asks, is it fishermen? Is it currents? How does that work?
We are working on a paper looking at that right now. We're looking at all of the data. So on those transect lines that we put down the bottom when we count the fish, we also count the trash. And we classify the trash into fishing trash and plastic and sometimes it's like construction trash. And it depends. It depends on how close to civilization the deeper reef is. So in reefs in the Philippines, where they're very close to a town, most of the trash is a mix of plastic, like small plastic wrappings of small things. And a lot of it is fishing lines, because a lot of people fish there, nets and just trash. I mean, we saw an engine from a boat. We saw a toilet seat. Right. There's a car that we found there. So the closer you are to civilization, the more human trash there is. The further you are, the more fishing trash there is. So the more broken fishing lines and broken anchors and broken ropes. We did an expedition last year to Trindade Island, that beautiful island close to Martin Vaz that I showed the picture of. That's 700 miles offshore in Brazil. Between that island and Brazil, there's a chain of seamounts that we stopped in one that's almost halfway between the islands. So 350 miles, no island there, nothing, in the middle of nowhere. We found a lot of anchors, broken anchors. So ships or fishing boats, they anchor there and they can't pull their anchor off anymore because it gets stuck to the reef. They cut their anchor. But in the process of doing that, it breaks a lot of coral. So we found a lot of broken anchors, a lot of fishing line in this seamount that is in the middle of nowhere, basically.
Yeah, yeah. Oh, Hootson just logged on and said hello. Hi, Hootson.
Hey, how's it going?
Hootson's the other diver you saw in that shark video. Okay, so this question is from Ace, and Louise, you're going to have to kind of like explain a little bit of this just for the mainstream viewers,
right.
Regular people like me.
Yeah.
I'm not sure if I remember it correctly, but is it true that corals in lower mesophotic ecosystems have a more upward morphology than in shallow reef?
Oh, interesting. I don't know if I can answer that because I'm not a coral specialist, but they do grow differently. So the question is, the corals, they grow differently depending on where they are on the reef or where they are depth-wise. They try to take advantage of light more. Mm-hmm. From the species that I can remember, it's the opposite. I think they grow more in an upward morphology on the shallows, mostly because they want to minimize how much surface they have exposed to the light. And in the deeper areas, if it's the same species that spams deep and shallow, the deeper it goes, the more plate it becomes, because they want to be a plate so they can absorb as much light as they can.
I see. Okay. Sorry that, yeah. Fish questions only, everybody.
I can
questions and
I
light
think I handle that well. Tim
I think
is
we
the
can
one
too.
I want to ask that.
Okay. Well, he said that he'll try to, but he's quarantining in
New Zealand, so it's gonna make
remote New Zealand. Yeah, with poor internet.
Have to do a dinner for him, not a breakfast.
Oh yeah, no, that would be good, but also he can't get online. He's like extra isolated.
Okay.
Him. Okay, last question from Becca. So, and this just seemed like a good place to kind of wrap up, because basically what she's asking is, she again was responding to the trash photo of the deep reefs, and she's saying when these deep reefs are both off the coast of inhabited places and also kind of in the middle of nowhere, and you see trash in all of these places, how do we actually protect them going forward? Like, what steps can we take?
Right. So in most of the places that we went to, including the last expedition to Brazil, one of the things we try to do is convince the local authorities to include those deeper reefs in marine protected areas. Mm-hmm. So the further you go, the more fishing trash there is. And the best way to avoid fishing trash is not going fishing there. So the best way is to kind of include those deeper reefs in marine protected areas. And amazingly, they have not been included. In Brazil, for example, there's Fernando de Noronha is the oldest and most successful marine reserve in Brazil. And it completely excludes, it stops at about 50 meters deep. So about 170 feet, the reserve stops. So all the Mesophotic, or most 95% of the Mesophotic reefs in Noronha are outside of the reserve. And we saw more fishing, more lines and more fishing related trash in those deeper reefs than we did in the shallow reefs that are completely protected. So the easiest answer in that case would be to extend the reserve, increase it a little bit to include at least a part of the Mesophotic reefs in it. So I think in terms of physical destruction and pollution and fishing, the best way to protect them is to include them in marine protected areas. And they haven't been so far, mostly because they're out of human reach. People think they're protected already and don't need protection, but they do.
Okay. Well, thank you so much. And there were a ton of questions we didn't actually get to, but we'll try to circle back and answer those in questions.
Right. And I'm in Twitter, I'm in Instagram. You can find me and ask me there, and I'll try to do my best to answer.
You want to say what your handle is?
Coral Reef Fish. Whatever.
Yes, and he's really fun to follow on Twitter. Everything from RAS Wednesday to I don't know. What else do you do?
Nasty gobies.
Nasty gobies. Ugh, don't get Louis started on nasty fish. Well, if you enjoyed this, and we hope that you did, so Wednesday and Friday of this week are going to be a 2-part nudibranch extravaganza, and
Nice.
Louise won't be impressed by this at all because he just only thinks fish are beautiful, but nudibranchs are sea slugs, and they're ridiculously pretty. They're like...
They're all right, too. I'd say they're all right. They're better than gobies, I can tell you that.
He really hates those gobies.
Definitely.
Yeah. So that's Wednesday and Friday morning, and then the following week we have everything from our chief of science, Shannon Bennett, who's a virologist doing life of a parasitologist. Everything from that to our planetarium director will be taking people on a tour of the universe. So keep coming back. Check the schedule that will drop in comments. And Louise, thank you so much. That was really fun.
Thank you.
Yeah. Take care. Bye.
Thanks for doing it. Thanks. Bye.